reaction, PCR, is a revolutionary method developed by Carrie Mullis in the 80s. PCR is based on using the ability of DNA polymerase to synthesize a new strand of DNA complementary to the offered template strand. Because DNA polymerase can add a nucleotide only onto a pre-existing 3'OH group, it needs a primer to which it can add the first nucleotide. This requirement makes it possible to delineate a specific region of the template sequence that the researcher wants to amplify. The PCR reaction requires the following components: a DNA template, the sample DNA that contains the target sequence. At the beginning of the reaction, high temperature is applied to the original double-stranded DNA molecule to separate the strands from each other. A DNA polymerase, which is a type of enzyme that synthesizes new strands of DNA complementary to the target sequence. The first and most commonly used of these enzymes is Taq DNA polymerase from Thermus aquaticus, whereas PFU DNA polymerase from Pyrococcus furiosus is used widely because of its higher fidelity when copying DNA. They can generate new strands of DNA using a DNA template and primers. They are heat resistant. Primers are short pieces of single-stranded DNA that are complementary to the target sequence. The polymerase begins synthesizing new DNA from the end of the primer. The general PCR cycle. A mix containing a template, primer, DNA piece, and enzymes must be heated to 95 degrees for separation of DNA duplexes. Then we lower the temperature enough for primers to anneal specifically to the template DNA. Then we raise the temperature to optimal elongation of the Taq or similar DNA polymerase, which is around 72 or 74 Celsius degrees. Then we repeat from top around 20 or 35 times with the consideration that less cycles gives less products and too many cycles increases the fraction of incomplete and erroneous products.
Now we are going to talk about the variants of the PCR. The colony PCR is a convenient high-throughput method for determining the presence or absence of insert DNA in plasmid constructs. Individual transformants can either be lysed on water with a short heating step or added directly to the PCR reaction and lysed during the initial heating steps. This initial heating step causes the release of the plasmid DNA from the cell, so it can serve as template for the amplification reaction. Another variant is polymerase cycling assembly, that is PCA. Assembly PCR using synthetically derived DNA is a very flexible technique for producing novel gene sequences. Single-stranded oligos or a mix of single and double-stranded DNA are used to produce longer genes of up to several thousand base pairs. The approach can also be beneficial for assembling constructs with the modulant elements such as antibodies. On the other hand, we have the droplet digital PCR. This is a method for performing digital PCR that is based on the water-oil emulsion droplet technology. A sample is fractionated into 20,000 droplets, and PCR amplifications of the template molecules occur in each individual droplet.
Primer design, we need to make the next considerations. The GC percentage must be in a range of 40 to 60%. The primer length will be between 18 and 30 base pairs. The alignment temperature is in a range of 52 and 60 grades.
First, a master mix was prepared at 3x in order to use it in the 3 samples that were used to run the PCR. That means the one with the genomic DNA, the negative control, and the positive control. In order to prepare the master mix, 30 microliters of 5X buffer were added. This buffer contained magnesium chloride. Then, 3 microliters of 10 micromolar DNA TPs were added. Following the procedure steps, the primers were added. First, 3 microliters of the forward primer were added. Then, 3 microliters of the reverse primer were added. Finally, the unity of the DNA polymerase and the 105 microliters of water were added in order to adjust the final volume of the mix. Once the previous steps were completed, the master mix was split in 3 parts in order to prepare the negative control, the positive control, and the mix with the genomic DNA. In the first microtube, 48 microliters of the master mix and 2 microliters of the genomic DNA were added. In the second microtube, the negative control was prepared, adding 48 microliters of the master mix and then 2 microliters of distillated water. Third microtube, the positive control was prepared, adding 48 microliters of the master mix, 1 microliter of water, and 1 microliter of the genomic DNA dilution. As we were working with very small volumes, and in order to verify that the preparation of the different solutions or mixes used in the lab, the volumes need to touch the entire solution and then with the micropipette leave the volume obtained. As shown in the figure, at the bottom of the gel, there are different blots that are out of the marker boundaries, but there is one that is darker and bigger than the other ones. Also, in the fourth row, there is a blot that is between 500 and 700 base pair, and is more visible than the ones that are in row 2 and 3 that are barely visible. There are presence of genomic DNA in the 3 samples prepared, because the binding pattern coincides in the first band. Also, the bigger band in the bottom of the row 1 may lead into the presence of DNA and DNTPs, not used material during the process. As shown in the table, the conditions of the primers are the best in order to use it with the target sequence. The table shows the conditions of the PCR in order to obtain good results. In order to know the stability and the good use of the primers, they were analyzed to find if they form secondary structures and also if they can form heterodimers or homodimers.
Various conditions are important while doing PCR. Knowing the polymers to be used is relevant to determine the condition at which the reaction should be held. To do a proper PCR, it's important to ensure that the template is not contaminated, as for the contamination with organic solvents could lead to the inhibition to the polymerase and affect the annealing kinetics. It's also important to design primers properly. We can see in the results of the electrophoresis that there are different plots which doesn't are in the marker boundaries. This indicates that the sample was less than the expected. This is probably a primer dimer, which also shows in the control band that was without substrate. This can be arranged with an adjustment of magnesium or primer redesign. Finally, the band of the positive control that also contains gDNA starts to blur at approximately 1500 base pairs, which presents a bigger or larger genetic information related with the presence of the X fragment of the gene and giving clues of a female sample.